All right, Tassa, how you doing?
I'm good. How are you doing?
Good. Thanks for being here today.
Yeah, of course. Thanks for having me. This is an honor.
Yeah.
So
How long?
yes,
How you just got off a plane?
I did. I just got off a plane. And it was a short 10-minute drive over here, and the weather is gorgeous. So I'm really excited about that too.
Yeah. I'm terrible at introductions, and I figured that out. So I'm just gonna let you introduce yourself and tell us where you're from.
Okay, sure. So my name's Tassa. I was born and raised in Colorado, and I did all of my education there. I am a molecular biologist. I did my graduate work in CU Boulder, and that focused on neurodegeneration, actually, and structured RNA. I then went on to do my postdoc at the Health Sciences Center in Denver, where I looked at actually nascent structured RNA and how RNA can lead to different diseases. So basically me and a couple of our science friends were in our postdoc, and, we're kind of done with academia, and we wanted to take what we knew how to do and apply it to do something useful to solve problems. And so we decided to start a biotech company, which was sort of the inception of Tumi Genomics. We kind of dabbled in a lot of different industries. We did some work on cancer genomics, viral genomics. We ended up creating and directing the COVID surveillance lab at the University of Colorado for about a year. And sort of underneath that, we were also working on viral pathogens in cannabis. And, over time, we realized that the need in cannabis for accurate diagnostics, for scientific education, for bridging that gap between the research and academia and what's going on in the industry was huge. And it seemed like a really great place for us to focus and make a big difference.
How long have you guys been doing cannabis viroid testing?
Yeah, so we actually started testing for Hop Latent Viroid, I'm going to say sort of informally, last spring maybe. So basically we were working on COVID and doing a lot of PCR on COVID, and we had a grower friend contact us and say, "Oh, I have this Viroid in my grow, and I know you guys are doing PCR diagnostics, and can you help us out?" And we're just a bunch of scientists, and we're like, "Sure, this looks really interesting." And viroids, they're horrible, but their biology is really cool. So we designed some assays, and she really liked it, and she started telling people about it, and it just kind of grew from there.
So Hop Latent Viroid, it's a viroid, not a virus.
Yes.
What's the difference?
Yes. So viroids are one of the simplest forms of life that we know of. So as opposed to a virus, which has kind of an outside protein coat, like sometimes you'll see it as like a diamond and then it has like these little legs. Viroids don't have any outside protein. They're simply a free-floating piece of circular genetic information. They're really, really, really tiny, but pretty infectious in plants.
Now, are they harder to track or treat compared to a virus?
So they're different, I guess would be the best way to put it. There are certain types of tests you cannot do on a viroid. So, like I'm sure people, because of COVID, have heard of like antigen tests. So those types of tests look for protein, and that they're really fast. With a viroid, you can't use tests like that. So you have to go with a molecular test because it's just the genetic information. So it has to be PCR or other versions.
So you have to do a full DNA PCR test to be able to see a viroid.
Yes, it has to be a molecular test of some kind. Yep.
So that kind of, as far as some of the questions that we were getting on IG were, is there a kit or a test that we could use in the field to test for Hop Latent Viroid?
Yes. So there are certain types of technology that you can do in the field. One of the ones that is employed most frequently is called RT-LAMP. So RT-LAMP is similar to PCR in the sense that it also finds the genome of the viroid, but it uses a specialized enzyme, so the whole thing can happen at one temperature. So you can put it in a machine and it just sits at like 65 Celsius. and it amplifies the viroid. And then you're able to see it. There's a lot of different ways to do it. A color change, or sometimes there's a glow, no glow. Those tests definitely have their place, and they're quick and on site. They tend to be a little bit less sensitive than a PCR. So I like to tell people when you use those on-site tests, at least the ones I'm aware of, it's really good at finding positives. It's less good at finding true negatives.
So just going back, what is PCR?
Yes. Okay. So PCR is a method where you use these little tiny things called primers, and they go into the tube and they look for a specific sequence of the viroid genome. And if they find that sequence, they use an enzyme called Taq polymerase. And so what that enzyme does is find the sequence, and then it makes billions of copies of that sequence. So even if you only have 1 or 2 copies in the tube, when you're done with the PCR, you have billions of copies. And so it's much easier to detect it. The nice thing about most PCRs is that there's a laser that reads every minute or so how much viroid is being amplified in that tube. And it can actually make a graph of how much there is. And so it can be quantitative. So it isn't just, is your plant infected or not? You can say, oh, this has really high viroid load or really low viroid load.
So a PCR kind of just amplifies the DNA and makes a small piece of DNA much larger.
Yeah, so it takes a small piece of DNA, and then it makes, of that one tiny piece, it'll make billions of copies.
Okay. So it gives you a broader test range so you can see it.
Yep.
And then you have LT lamp, which is a different technology, right?
Right. So it's RT lamp.
Okay.
Yes. But so RT lamp, it's similar in the sense that it also finds a piece of the genome and it makes billions of copies, but it uses a different enzyme. And this enzyme is able to function at one set temperature. So normal PCR requires the cycling of temperatures, which necessitates like a big expensive machine. Whereas RT lamp can all be run at one temperature, so you can just do like a little heat block or something. So it's also an amplification technology, but slightly different.
So going on the product to have in the field, is it expensive to have something in the field that will be able to see Hop Latent Viroid or test in the field?
So there's several different tests. Some of them are not terribly expensive, and they're not even that complicated to run. So I don't know if people have seen those tests where you have a tube and it's pink and then it'll turn yellow. Mm-hmm. So that's RT lamp. And those aren't expensive or difficult to run. There are also like handheld type platforms that you can buy. I think Agdia has one that you can even get some sort of quantitative information. That's a little bit more expensive because it's a machine. We're actually working on an on-site platform too. That
was my next question.
Yes, yeah. So there's a lot of potential issues with RT lamp because it can be non-specific. It can throw false positives, and because of that, it's not terribly sensitive. So what we've actually done is changed the chemistry of RT lamp to improve some of the problems. So ours, it has an internal control. It's more sensitive. It's less subject to human error, and it's high throughput. So it'll be able to do 93 samples at a time.
Wow.
Yeah.
And when is that going to be ready? What are you thinking?
So we're hoping to have the beta version ready in the next few months. And then the full version, which comes with a full analysis machine that will feed directly into a database, that's probably a little farther off.
That's super exciting.
Yeah, no, we're really excited. We developed it for COVID, and it was being used for that. But I think it can have a lot of applications in cannabis too.
Yeah, that's awesome. So what is DNA sequencing?
Yes. So to understand this, I might have to delve really shortly into a lesson in biology. I'll try not to be super boring. So DNA is basically the instruction manual inside your cell that tells the cell everything that it needs to know to do whatever it needs to do. And so just like an instruction manual or an encyclopedia, it has DNA has letters, but instead of 26, like the English alphabet, it just has 4. So A, T, C, and G. And it's the order of these letters that give the meaning, just like in language. It also has punctuation marks and capital letters, and it even has sentences. So sentences in DNA sequence are called genes, and they're like one complete unit that can be understood. And so when you get a DNA sequence, you're getting the order of those letters, with the idea that you can then find meaning in that by identifying where the sentences are and what they're trying to communicate.
And with the in that, and that's pretty much genomics is testing and looking at that sequence.
Yes, trying to understand what the order means, how the sequences interact with each other, how they're expressed in the cell.
Are you familiar with anybody taking those DNA sequences and, applying intellectual property towards them? I mean, what do you know about that? Because I was watching some stuff on DNA sequencing and intellectual property with cannabis.
Mm-hmm.
And like the first question that comes to my mind is like, how when it's not federally legal?
Yeah. So I will preface this with I'm not a lawyer, so take it with a grain of salt. So I do believe that you can patent cannabis even though it's not federally legal. In order to get a patent, you have to prove that whatever the strain is that you've created is unique, it's non-obvious, it's not available in the wild, and you've created it. So one of the advantages to DNA sequencing is that you can sequence the genome of some, new cool strain that you've created, and then you can identify genetic markers that track with whatever that trait is that you think is unique and non-obvious. And that data can help you with your application because you can claim that this is the genetic basis for it, and you created that.
So just breeding two genetics together and creating another DNA sequence, right? Because that's what you'd be doing.
Mm-hmm.
You could take that and possibly patent that.
Yeah. So example, if you put two plants together and you create a new plant that has some sort of unique cannabinoid or some really cool growth pattern or whatever, if you can find the genetic basis for that and you can prove that most of the cannabis plants out there don't have that allele or that sequence in their DNA, then you can make the case that you've created a new strain that is non-obvious and unique, and then you can patent that.
Are they doing that a lot in vegetable crops that you know of, or?
Yeah, so there's a lot of patents in normal agriculture for new strains, or a lot of times you'll have two genetics and you cross them together and you create something called an F1 hybrid, which is a really robust plant, and you can sort of patent that process too.
So can anyone patent genomes just off the street? Hire a IP attorney and just.
So my understanding is as long as you can defend that you have invented it and created it and that it's unique and non-obvious, then yeah.
It seems weird when it's like a natural plant.
Yep. Yeah.
I mean, it seems like, how can you patent that?
Right. Exactly. Yeah. So you're patenting like a unique
Trait.
Exactly.
Okay.
Yep.
With genomics, can you map lineage and see where certain cannabis plants have come from?
Yeah. So what you can do is you can take a cannabis plant and you can sequence the DNA and you can analyze spots in the DNA that tend to change. So they call these spots SNPs or indels. And you can find all the places that it looks different in the DNA sequence. And then you can compare it to a reference genome or another cannabis strain. And depending on how many of those differences they have from each other, you can say whether or not these two strains are genetically really close together or genetically farther apart.
And telling you where that plant came from a certain region, in its lineage.
Yes, as long as there's enough genomic information or genomes available for the different regions and different types of lineages, you can fit it in there.
with breeding cannabis plants, can genomics pick certain genetic expression?
So that's a complicated question. Right now, the genomic space in cannabis is in its infancy. The genome was just sequenced, in the last couple of years. And even though it's sequenced, there's still parts of it that aren't done yet. I think 10% of it or so is missing, depending on which reference genome you look at. And something like 10 to 25% of the reads can't be mapped. So in order to predict genetic expression, you not only have to have the sequence, you have to be able to find the genes or the sentences in that sequence, and then you have to be able to know what they do, and how different changes in each sentence is going to impact how the plant looks. And that's a really complicated process. And the cannabis genetics space is getting there, but I don't think it's quite there yet.
Is there one like firm or lab that kind of leads the charge of, sequencing cannabis, or does everybody come together as a whole?
So there are academic labs that are doing sequencing. There are industry labs like Medicinal Genomics that are doing sequencing, but they're also publishing papers and making some of the genomes publicly available, which is really helpful for advancing our understanding of the genome.
So they could say, hey, we found this gene, and we visually look at the plant, and the plant seems to grow shorter or have wider leaves and hardier.
Yeah. So what we're able to do in cannabis is something called forward genetics. So in forward genetics, you have a plant that has some sort of interesting, phenotype like flower or whatever, and you want to understand the genetic basis of it. You can sequence the parents, and then you can make a cross and take, hundreds of little babies and then sequence all of them and then let them grow up and find which babies have that trait, and then go back to the DNA sequence and find changes that correlate with the development of that trait. What's harder is reverse genetics, which is where you look at the sequence and you try to use it to tell the future. Like, this is going to mean that this is going to happen. That's much more difficult.
That this plant will grow taller or shorter or possibly be disease resistant.
Yes. Yeah. So a lot of that is, it requires a much more in-depth understanding of not just the DNA sequence, but how that sequence is interpreted by the cell, and how that sequence interacts with the genetic background of each of the cultivars. And so it's a much more complicated question.
Do they are you familiar with any genes that they've found that are more resistant to powder mildew or botrytis?
Yes. So they have been looking at this. I think that they found something like 80 or more genes that can correlate with powdery mildew resistance. Medicinal Genomics did do one study that was pretty nice, where they found one specific gene that they thought could be the cause. They took that gene out and they expressed it in a bacteria, and they showed that the protein that it was making inhibited the growth of Fusarium. And so that's the closest I've seen to actual causation so far.
Would you be able to breed that gene into other genetics using like CRISPR or something?
So theoretically, you could do that. Whether or not that specific gene is sufficient in the plant in vivo to cause powdery mildew or Fusarium resistance isn't known. And you have to remember that if there's, something like 80 different genes that are controlling the resistance, how that gene is going to interact with the whole pathway to result in resistance in the plant is a much more complicated question. But theoretically, if scientists could find enough genes that confer resistance, they may be able to create resistant plants using CRISPR.
Is CRISPR GMOs, or is that a different?
So the
I mean,
definite, yes, it's hard. That GMOs is a difficult thing. I believe that if you induce a gene that normally exists in the plant, it's not considered a GMO because you're sort of speeding up a natural process. If I were to throw in like a gene from a fish or something to make it cold tolerant or whatever, then that sort of crosses the GMO line. But it's fuzzy.
So CRISPR can be qualified as GMO.
It can or can't, depending on what you're doing with it.
Yeah. So I heard that we met because I used Tumi to find Hop Latent Viroid in my spot in Nevada.
Okay.
And I had sent in some samples, worked with Patrick.
Yeah.
And got a bunch of samples back, and we had taken 5 cuts off of one plant.
Mm-hmm.
And every single cut came back as positive of Hop Latent Viroid. And I was like, wow,
yeah,
this, that we're able to weed this out. And I've been in a lot of facilities,
mm-hmm.
All around the United States. And a lot of the times, I was in one in Vegas and walking through the veg room, and I'm telling the guy, hey, I think you got Hop Latent Viroid here.
Yeah.
He was, no, I don't have Hop Latent Viroid. I don't know what that is. I mean, a lot of people don't know what it is.
Yes, exactly.
Yeah. What is Hop Latent Viroid?
Yeah. So Hop Latent Viroid is a tiny little free-floating piece of genetic information that it actually was first found in hops. That's hence the name Hop Latent Viroid. The reason they call it latent isn't because it can lie dormant, like hidden in your plant, but because when they found it in hops, they couldn't find any effect for it. So they decided to call it latent, which really just means asymptomatic. That actually turned out to not be such a great name because it isn't totally asymptomatic in hops either. It can mess up like oil and acid levels in the flower, and some cultivars are super sensitive. And then of course in cannabis, it's not asymptomatic at all. So it was just a poor name choice.
So it's not latent.
It's not. So it's a really confusing term. So in science, there's something called lytic and latent. So this is a cycle that some viruses do, especially like retroviruses, where they can intercalate into the DNA and then they just like hang out. And they're kind of quiescent. They don't replicate or make new viral particles. And then when conditions are right, they sort of wake up and they enter a lytic cycle where they're doing a lot of replication. Hop Latent Viroid doesn't do that. So it doesn't actually technically have a latent phase. If it's in the cell, it's trying to replicate itself.
Now, what are the growth characteristics that you've witnessed? I know do you spend a lot of time in the garden, or do you mostly just in the lab and running Tumi?
So I definitely spend more time in the lab than in the garden, but I've definitely seen my share of Hop Latent Viroid infected plants and talked to a lot of growers. So I do know that the symptoms can be really pleiotropic. So which means like super varied. So you can have a plant that's either really severe or, so subtle that you don't even see it. Common symptoms are, your plant will be slow growth. You can have malformed leaves, leaf chlorosis, brittle stems. Sometimes you'll see like sort of a horizontal grow, or the leaves will overlap a little bit. But of course, the most insidious phenotype is that it totally destroys the flower. And
at that time it's too late.
Yeah,
you've
exactly.
already 3-4 weeks into flower, and then you're seeing what's coming out of there, and it's not.
Yeah, it's a huge disappointment.
Yeah.
Yeah.
It takes a really trained eye to see Hop Latent Viroid.
Yeah.
I worked with a few growers at my spot, and I'd walk through the veg room, and I'd be like, hey, you got HLV here, you got HLV here, and they're like, what do you mean? Like, I was like, you kill this plant, kill the plant, throw away the scissors.
Yeah. Yeah.
What I seen is kind of, instead of the branching, first signs that I look for, instead of the branching growing up at more of a direct straight angle, you kind of see this flow out sideways and then up.
Yeah. Yeah.
That's one sign. They come out sideways and then they go up instead of shooting straight off the main stem. And then also you kind of get this slight curl of the leaves start to kind of come together a bit.
Mm-hmm. Yeah.
And you'll see that. And then that's in veg, and those are the first signs that I really look for. But in flower, it's like, you've already filled up half your garden with HLV, and then you're flowering out, and you're like, do I even keep this going?
Right. Exactly. Yeah.
How does HLV spread in the garden?
Yeah. So HLV spreads via mechanical damage. So unfortunately, the biggest offender for HLV spreading is us. It's very efficient at spreading on unsterilized tools. So if you're trimming a plant that has HLV and then you use those same scissors on your next plant, you've just inoculated the next plant with HLV. It can also spread to a lesser extent on trimmers' hands or maybe even their clothes, especially if you get sap or something and you're not changing your gloves or cleaning them with bleach before you move to the next plant. That's a potential spread there too.
Even you don't have to just open up the plant. Say we take a cutting, you're opening up the tissue of the plant. You can spread it just by touching.
So you have to have mechanical damage, but
Okay.
it doesn't have to be like catastrophic, right? So if you're trimming and you get a little bit of goo on your gloves or whatever, then it's on your fingers, and it's a potential possibility if you're not sterilizing yourself between each plant that even your hands could spread it.
Can bugs like thrips and mites spread HLV?
Yeah, so that's a good question. They have looked at this question in hops. They did an experiment a long time ago where they took like 1,700 aphids and they threw it on a really infected hop plant, and they let them munch away, and then took those aphids and threw them on healthy hop plants. And they didn't develop Hop Latent Viroid. And so the conclusion from that study was that, at least in that setting with hops, the bugs weren't transmitting it. That doesn't mean that it's couldn't happen in an indoor grow setting with cannabis plants. So it's kind of an unknown right now.
Have you seen some genetics not susceptible to Hop Latent Viroid? They have Hop Latent Viroid, but they're still getting good yields. They're still getting terpene production. They're still getting everything, and it still has Hop Latent Viroid.
So I've never seen that, but that doesn't mean that it doesn't exist. And I have talked to growers who, their plant is definitely positive according to the PCR, but it seems everything seems fine. So the fact that there can be plants in hops, the cultivars that are basically asymptomatic, it seems possible there could be some in cannabis as well.
I had a question on Instagram. The question was, does HLV change the DNA or the RNA of the plant?
Yes. So this is a complicated question. What HLV can't do is get into the DNA of your plant and kind of live in there and be transferred from plant to plant. So it can't be like part of the genome of a particular strain. Once it's in the plant, part of the mechanism for making the plant sick does involve changing gene expression. So it can change something called the epigenetic control of genes in the plant. And it can also impact the way that the RNA is processed and expressed. That type of change is unlikely to be transferred to plant, like if siblings or in the seeds or whatever, unless Hop Latent Viroid is also transferred into the seed.
There is a possibility if you do breed two plants together that one of them has Hop Latent Viroid or both of them, that it doesn't make it to the seed?
Yes. So the seed transmission, so that question is kind of up in the air in cannabis. There are studies in hops claiming that it's actually pretty rare. I think they put it about 8%, and they said that the level in the seed was pretty low. We've done some experiments testing seeds, and they're definitely come out positive, and they come out very positive. We're actually running an experiment right now with a grower who ended up in that situation, and he's like, I have all these seeds, what do I do? And I'm like, well, if you're not going to use them, send them to us and we'll PCR each one and tell you what the percent transmission in seed is for cannabis. So we're working on that question right now.
So you don't have the data back from that.
I don't,
Yeah.
not yet.
So you actually test each seed. You put it in and you grind it up,
Mm-hmm.
and then you're testing that.
Yep.
HLV, you're able to see it by the genetic marker.
Yes. So we detect it by PCR. So
Okay.
each one of those becomes a PCR reaction, and then we can come up with a percent.
Regarding tissue culture, have you seen tissue culture be able to cure HLV?
So, there is actually some published evidence in hops that tissue culture can cure HLV. It's not 100%. So a lot of it depends on when the plant was infected and what cultivar you're doing it on. So it can go from 0% effective to 50% of the time it works. I do know that there are some companies that we test for, and we also test for the people sending their tissue culture clones there. And so I can tell you that at least for the companies that I've had experience with, they are successful at eventually ridding a really important cultivar of Hop Latent.
But it could take multiple tissue cultures to get there.
Yes, yeah. So when you take the tissue culture, not every single one is going to be Hop Latent Viroid free. So you have to test each one and find the ones that come out negative.
Yeah. When you're pulling, when we took our tissue tests to do the Hop Latent Viroid, you had us pull 5 samples.
Mm-hmm.
Why did you do that?
Yeah. So it turns out that the viroid load throughout the plant can vary a lot. So we actually ran a little experiment with a different grower to see how much it varied. And we found that if you sample, from top, middle, and bottom, it can vary by up to 560-fold, depending on where you take that sample. So if you only grab one spot on the plant and you're unlucky enough to grab that spot where the load is really, really low, you might miss an infection. So it's better to take multiple samples.
Now, should they take it off multiple branches around the plant?
Yes, I would. I would try to distribute out to as many different places as you can, and then do multiple branches. And you can put it, at least for our test, you can put a little tiny clipping all in the same tube, and then it all gets processed together.
So that would it be easier to take cuttings from maybe a brand new baby or new vegged plant instead of a big mom?
Yeah, so probably you'd have a better chance of catching it if the plant is much smaller. Most people are in the situation where they have a big mother and they want to clone, like you take like 100 clones or whatever, and they need to know if it's infected. But yeah, probably catching it in a smaller plant might be a little easier.
Yeah, I think so. I mean, yeah, a lot easier because instead you have one main top. You take that one top and right away.
Yeah.
I mean, that's most of the tissue of the plant.
Yeah.
So it's probably actually better if they did want to get the big mom tested, just take a few cuts, root them out, transplant them, veg them for a week or two, take the first initial cut and send that in for testing. Wouldn't you think?
Yeah. So, I mean, I guess it just depends on, where you're at in your rotation and what you need to get started. I guess there's always a possibility of missing it. That's the hard part. I would, if you are going to take that route, I would probably test over several weeks that same plant just to make sure before you grow that one up.
So test it a few times.
Yeah. More than once.
Yeah, not just one time and you're good.
Yes,
keep
exactly.
going.
Yeah.
What can growers do to prevent HLV in their gardens and from spreading?
Yeah. So I would say the most important things are to enact a really strict standard operating procedure around tool sterilization, and probably personal protective equipment for your workers. You obviously need to also come up with a pretty rigorous testing protocol. I know it's kind of a drag, but if I had to grow, for example, I would test all of my mothers, every 3 to 6 weeks because it's just not worth the economic loss that you could suffer if you had a rampant infection in your mom room, for example. Um, you know, because information is always better than just hoping. Like I've had growers tell me, well, we're just hoping it's okay, or we have our rosaries, and I'm like, it's always better to just go ahead
Yeah.
and test. Yeah.
Now, when we were cleaning our utensils and cleaning our tools, just naturally we used isopropyl alcohol.
Mm-hmm.
And we thought, hey, alcohol kills everything.
Right. Yeah. A lot of people say that.
But
yeah, unfortunately, not only is alcohol not effective, nucleic acids are really stable in alcohol. So they're really happy to be dipped in alcohol. There's actually a lot of studies about what works and what doesn't. And so because I get asked this question so much, the things that do not work, alcohol doesn't work. Alcohol followed by flame doesn't work. Torching your scissors does not work. A lot of the cleansers that are based on hydrogen peroxide or acetic acid, those don't work. Ammonium cleaners don't work, like Lysol. UV light does not work. I know, right?
What?
I know.
I'm tripping right now.
So the things that work in every experiment are Vircon S, 2%. I do warn people the 2% is really critical, because if you do 0.5% or 1%, it doesn't work. Or the thing that I like to recommend is good old household bleach. That works every time in every experiment they've done since like 1969.
How long do they need to leave it in bleach? I'm still tripping. Hold on, flame doesn't work?
Nope. No, unfortunately, I know it's crazy, but it's not 100% effective. So
is that just because it's a viroid and not a virus?
Yes. So because it's just that free piece of genetic material, it's a lot more resistant to most of the things that you think of that would like sterilize something. So yeah, viroids are tricky. They're a lot trickier than other stuff.
Yeah. So the whole time that I've had my cup of alcohol and we're putting our scissors in our alcohol, it's actually helping HLV spread because it does better in alcohol.
Yeah, it's mostly like a little HLV culture that you've got going on in the beaker that you're spreading around to the other plants. Yeah.
Oh my gosh.
I know. It's scary how many people tell me, "Oh, it's alcohol." And I'm like, "No, that doesn't work at all." So,
how long does it need to stay in the bleach?
Yeah, so you want to use somewhere between 10 to 20% bleach, and 30 to 60 seconds is sufficient in order to deactivate the viroid. Some people say like 4 hours or overnight. That'll work, but your tool is going to be like a puddle the next day.
Yeah.
So
30 seconds, you're cool. Just have a jar there, throw the scissors in, and kind of each branch that you're taking.
Yeah.
Maybe clean in between.
Yeah, if you have, a couple scissors and you have them in the bleach and you grab one and clip and then throw it back in there and then grab the next one. And as you're moving from plant to plant, if you have like some bleach spray, you can spray your gloves real quick before you go to the next plant to kind of make sure that you don't have anything on your gloves too.
And spray surfaces that you're going to be cutting clones on.
Yeah.
So it doesn't spread.
Yeah, and do make it up fresh every day because the concentration can fluctuate if you let a dilute solution sit around for a while.
This is game-changing information.
Yeah, no, I get to ask this question multiple times a day, so I'm excited to just get that out there and help people out.
What are some of the other questions that you're asked that come off the top of your head?
Let's see. I get asked a lot how the viroid moves around the plant, and that generally involves it hijacking its normal biological systems that transport RNAs and proteins around the plant. I get asked what part of the plant has the highest level of viroid, and that seems to be really variable. There were some experiments done in potatoes or tomatoes. It was actually tomatoes, where they inoculated a plant with a different viroid that is common in tomatoes and potatoes, and they actually found that the viroid was easily detected above where they inoculated the plant, but not so much below. And then it's really common in the roots. So I would love to do that experiment in cannabis and see if it's the same for a cannabis plant. Probably one of the scariest routes of transmission that we have inklings occurs is through the water. So we can definitely find Hop Latent Viroid in the roots. We can find it in the growing medium. And we actually had one grower submit the little water emitter with some tubing attached. So we just cut off a piece of the tube and we used it as the template for the PCR. And we were able to detect viroid inside the water tube. So when people have Hop Latent Viroid and they have a common water source, they need to be thinking about that too.
So DWC and recirculating systems.
Yeah. Yeah. Or, those clonal trays or like a flood and drain, all of those could be potential ways to spread it.
And you tested the roots and it had positive Hop Latent Viroid inside roots.
Yes. Yeah. And it's actually, if you look at in hops, what tissues have the most viroid, roots are actually pretty high for concentration.
That's super scary for anyone that's using DWC.
Yeah, it is pretty scary. Yeah. And the scariest part is that viroids are really stable in water. So the potato spindle tuber viroid, which is really similar to Hop Latent Viroid, is stable for 7 weeks in water. So it's, you really have to think about your water source and where that water was before it's watering your healthy plants.
Since you've been in the cannabis business, have you kind of seen a progressive to it getting worse and worse?
It's hard to say if it's getting worse or people are just becoming more aware of it. When we first started doing this, there were a lot of people that were like, what is the Hop Latent Viroid? I've never heard of that. I don't know what that is. But I think that awareness of it is becoming more common. And so we're getting a lot more people submitting samples. And we're seeing it, kind of all over the United States. So it's everywhere.
Do you have any other advice on seeing it or not spreading Hop Latent Viroid across your garden, other than bleach and strict SOPs of cleanliness?
Yeah. So I guess my advice would be to sort of, for lack of a better way to say this, raise the level of anality for cleanliness like a little bit higher. I would recommend to not let any new genetic into your facility until you've thoroughly tested it. Even if you get it from a really reputable source, they might not even know that it's infected, and it's just not worth the risk of bringing something infected in. I would treat your mom room like a temple. Don't let anyone in there unless they know how to sterilize themselves. They understand how it's transmitted, and they have the appropriate amount of respect for how bad Hop Latent Viroid can be. And I would routinely test, even if everything is great and everything comes back negative. It's better to always be monitoring that situation so you can catch it early versus waiting until your plants flower and you're like, what? And then, you've got a lot of backtracking to do to fix the problem.
Yeah, then you're chasing your tail
exactly
the entire time.
Yeah. And, for a lot of growers, losing like a whole harvest is kind of, it's economically devastating. So it's worth the investment. The risk payoff ratio is pretty good for testing. I think.
You guys are saving people's businesses.
Yeah. And I know we have a testing lab and whatever, but you don't have to test with us, but people should test. Pick a lab you like, make sure they're doing good science, and get your stuff tested.
Yeah. No, when I was that story I was telling you where I was at in Vegas and seeing that kind of growth, the first question I asked him is, how long have these plants been here? Because none of your other plants have Hop Latent Viroid. Just this small, 8x4 section of plants that you have here definitely all have it.
Yeah.
And he's like, oh, I just got those from my boy like 3 weeks ago. I just brought them in. I'm like, hey, get these out of here right now. Like just it to come into somebody's garden and to just tell them cut all these plants down. You gotta have some balls,
right? Yeah.
And I'm just like, look, cut these things down. And he's looking at me like, this guy's tripping, But it's cool. I got a call about 4 weeks later, and he was like, look, I sent him in and I got it tested, and dude, you're right. They all have Hop Latent Viroid.
Yeah. No, I
And,
mean, you saved that guy for sure.
and because his entire rest of his garden was perfect.
Yeah.
Nothing. And the thing is, it could all start right there with that choice of bringing some plants in from your boy or whatever. And it just ravages your business and devastates you.
Yeah. Exactly. It's like I said, it's just not worth bringing something in unless you just test it. Just test everything before you put it next to all of your really important genetics.
Yeah. That's probably like number one advice. Don't bring anything in without being tested.
Yeah. 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 And I hear a lot of people say, oh, but, a lot of people recommended this or that or whatever. And I'm just like, test it anyway.
That's exactly why we do these podcasts.
Yeah.
Because everyone has their own opinion or their own views or what their boy taught them or this since they've been doing it. And with here, it's just a clean slate. We ask people like you, the CEO of Tumi Genomics, and get the right answers to the questions instead of what your boy, told you.
Right. Yeah. Yeah. There's a lot of crazy stories and misunderstandings out there. So it's nice to set the record straight so that people, can start fixing the problem or avoid it altogether, hopefully.
Yeah. Are you guys testing any other pathogens or working on any other testing for other pathogens?
Yes. So we so besides Hop Latent Viroid, we're also actively doing Fusarium oxysporum. And we're hoping in the next month or two to have released a full viroid and full kind of like fungal mold panel so that, if either your plant turned out negative for Hop Latent Viroid or you don't know what's wrong with it, you can kind of order something a little bit more broad to identify it. We're also sort of in the long term trying to move into next generation sequencing type analysis, looking for viral pathogens so that we can do it in a more agnostic way. Because the number of pathogens that I predict that we're going to find in cannabis is probably just going to explode. There's a ton of pathogens in hops, and there's no reason, because those two plants are so genetically similar, that they wouldn't cross over into cannabis. And so I think along with looking for stuff that you already know about, it's important for the industry as a whole to start thinking about what's coming and try to characterize that stuff too.
I mean, there's stuff that's out there. It's probably there that we don't even know yet.
Exactly. I'm sure that there are plants out there infected with stuff that nobody's looking for. And I guess one of the scarier things is that, once you have a crop that's so geographically spread out that has a viroid or a virus or whatever, it's just a short distance to it spreading to other agricultural crops like, staples like carrots or lettuce or whatever. And so it's even more important on a big agricultural view to be monitoring cannabis and treating it like a real agricultural crop that needs to be characterized with its pathogens and biology and stuff.
What is Fusarium? And
yeah, so Fusarium is it's a fungal pathogen. It's probably one of the worst. Fusarium oxysporum. There is no cure for it. It infects through the roots, and it usually causes like dampening off, and eventually plant death. It's really infectious, so it can spread around through the water, through on hands, just like Hop Latent Viroid, through the air if there's spores, and so it's another big scary one, especially with hydroponic systems that people should be considering.
So any kind of DWC or recirculating, you'll spread it throughout your whole garden.
Yes, yeah. So if you have a plant hooked up into that common system that has Fusarium, chances are you're gonna gift that to the rest of the plants too.
Are you familiar with any water treatments that kill Fusarium?
So I don't know of any water treatments. I do know that there are some harsher treatments that you can put on plants to help with Fusarium, but they're not really recommended for cannabis because it's, people are going to eat it later and they're pretty toxic. And so in cannabis, there really isn't a treatment that you can put in there that's safe, that'll take care of the problem.
So you're saying Fusarium is not curable. So what does a cultivator do to get rid of it?
So it's very similar to Hop Latent in the sense that you got to find it and you need to destroy the plants that have it. We've worked with some people that claim that they can take a plant and like continue to clone from the top and kind of clone away from it, and they've had success with that. Um, but your best bet is to just destroy the plant and sterilize everything. Bleach works for that too, and try to get away from it like that.
When you're getting a test for Fusarium, you're doing water samples?
So we can do water samples, root samples, soil samples, kind of whatever you think the problem is. We usually do root, and then if people find it, they're like, okay, where's this coming from? And they'll try, sending in a little bit of rock wool or a little bit of water to see if they can figure out how it's getting into their grow.
And you're able to get away from it. Some of your clients have said that, oh, we've cut from the top and gotten away from it. But is Fusarium in the tissue as well?
Yes. Yeah. So it starts in the roots, but then it can spread to the rest of the tissue. And so if it's really bad, it's going to be, impossible to clone away from it. But like I said, I haven't read any literature on it, but I have heard from people that test with us that they had success doing it like that.
What are you seeing on inbound percentages of Hop Latent Viroid to Fusarium?
So we test a lot more for Hop Latent Viroid than Fusarium. So we don't have really good percentages. We've definitely done tests and grows that had both, which was really bad. But
yeah. Well, I'm sure there's a lot, to be honest with you.
Yeah. Yeah.
Because it's you can't see, a lot of growers just can't see either of them.
Yes. Yeah. They're hard to spot and it can initially also be kind of asymptomatic. So, and then they're hard to get rid of. So
is Fusarium a lot harder to detect visually in the plant compared to Hop Latent Viroid?
It kind of depends on if you know what you're looking for. I think eventually it gets really severe and it's really obvious, but I think at that point it's too late. And so the trick is to catch it before it's really bad and then it spreads all over the grow. And so at the moment that you want to be detecting it is when you can't really see it anyway.
That's scary.
Yeah, it's
I'm going to get
scary.
some Fusarium testing going.
Yeah, yeah. I mean, and it's a lot more common in like hydroponic grows and stuff.
Like rock wool.
Yeah. Yeah, we see it more frequently, in Colorado because everything is like super indoors because it's really cold. But yeah, it can be just as scary as Hop Latent Viroid actually.
And it can spread just as fast.
Yep, it can spread pretty fast.
So that's super cool. Well, I appreciate you making the trip out.
Yeah, no, of course. This was a super honor, and it's really exciting to talk about this stuff and help people.
Yeah.
Figure it out.
Save you a lot of time on the phone.
Yes, exactly.
I think
so
you could kind of tell your clients, hey, just listen to this podcast. You got all the information.
Right. Exactly. Yeah. Especially with the tool sterilization one. I'm hoping that everybody hears this and everybody changes that, because I think just that one change alone is going to make a huge impact.
And the consciousness of Fusarium, as far as I'm really concerned about this now,
Yeah. Yeah. I mean, and like that, there's other pathogens too. So it's just something to be cognizant of and know that, you have to be aware of it. You have to be careful with your tool sterilization. You just have to be thinking about it, and then you have a chance of avoiding it.
Before we end, what are some of the things that you're excited about that Tumi's working on that you'll be releasing soon?
Yeah, so I would say the most exciting thing for us would be our on-site testing platform. Mostly because we've worked really hard on the molecular basis of the test to sort of change the chemistry so that a lot of the problems that people are currently having with on-site testing, it can solve that. You don't have to open the tube after you've amplified material. It's a visual readout, so you can see it. It has an internal control, so if a plant is negative or you just totally messed up the test altogether. And it's high throughput, which I think for a lot of grows, a lot of the current solutions just aren't feasible if you have 1,000 plants to test. So we're really excited to bring that out, and hopefully people can get some on-site information about their plants.
Where will they be able to? Will they be able to buy it through their local hydro store, through your website? Like, how would they get their hands on it?
Yeah, so initially it's going to be through our website, and then hopefully we'll be able to distribute it out and get a farther reach. But that remains to be seen.
Yeah, I think it'd be awesome if you guys could distribute to hydro stores.
Yeah.
Just because a lot of the foundational cultivation for the licensed facilities is the bedrooms and garages and basements.
Yes.
And if we can help make cultivators conscious in the basement, we can then help, focus on the licensed producers and spreading it kind of where the source is.
Yes.
It's coming from.
Yeah.
So that I think getting it out to hydro stores and giving hydro stores an opportunity to sell it to their, small and at-home growers is going to be game changing.
Yeah, no, we would love to make it accessible to everybody, and the only way to make a dent in this is to address it on every level, like you said, from, the basement growers all the way up to like the huge vertically integrated growers. Because if we leave somebody out, then you're just leaving a reservoir for Hop Latent Viroid or any other pathogen to just rear its ugly head again.
Yeah, exactly. I just want to thank you for changing my garden. Because you're in there and you have to kind of it's a guessing game the entire time, and having a company like yours that you can go to and get a definite answer, like this is what's going on, gives you that ability to clean it out and get rid of it, and pretty much changing the industry, so it's awesome what you guys are doing. Super excited about the at-home test. Super excited for everybody to be able to get their hands on it and do it at home, in home grows and licensed facilities. I think you guys are going to change the industry for sure. Appreciate you making the flight out to California.
Yeah, no, and thank you so much for inviting me. It's really exciting, and I like science, so it's fun to talk about it, and it's fun how many people are interested in it. So this was a really great opportunity. Thank you.
You're in the right industry now, or in the industry now, where you got a bunch of little scientists at home, with their grows and doing their thing, and everyone's that's what I loved about it most. I love to grow plants, since I was like 9 years old.
Yeah.
So it's cool that we get to talk about it.
Yeah, no, and I'm surprised how many growers are excited about doing experiments with us. So you're like, oh, let's do an experiment. And they're like, yeah, let's do it.
Yeah.
So I'm like, this is great. Just a ton of scientists. Yeah.
Appreciate it. And let's wrap it up.
All right. Sounds good. Thank you.
Thanks, Tassa.